I would like to begin by acknowledging and paying respect to the traditional owners of the land on which we meet, the Gadigal people of the Eora Nation. It is upon their ancestral lands that the University of Sydney is built, and as we share our knowledge, teaching, learning, and research practices within this university, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. Welcome everyone to the third Sydney Nano Meet the Author event. This is an event run by the early and mid-career researcher ambassador program, where we like to showcase and highlight some of the amazing work done within the Sydney Nano community. And we've had a couple of speakers over the past few months. You're able to catch those on YouTube. If you look at Sydney Nano Meet the Author, they should be there. This is an opportunity for a speaker to talk a little bit about the technical side, which is fun, perhaps on a higher level. So it's aimed at a broad audience, but also it gives us an opportunity to talk to the man or the person behind the research. So you are welcome to treat this as a kind of interactive podcast. We're a relatively small community. We can jump in and ask questions. This is an informal discussion, a little bit about science, a little bit about people, a little about the journey we are all on. So I welcome you all to jump in and treat this as an interactive experience. So I'm Alessandro Tunis, by the way. I am at the School of Physics, but also, of course, a member of Sydney Nano. And today we have joining us Nicholas Hunt, who is a lecturer based at the Concord Clinical Schools at the Faculty of Medicine and Health here at the University of Sydney. He's an early career researcher developing nanotech for targeted drug delivery of therapeutic agents for metabolic disease and diabetes, and I believe he'll be telling us all about that today. His team has developed intellectual property around oral peptide administration. He's part of the Sydney Spark program. He's part of the Nano and Sydney Nano Health Network, and maybe he'll be talking a little bit about that. He co-chairs the Nano Pharma cluster. He is a secretary of the Faculty of Medicine and Health EMCR committee, and he received a Sydney Nano Publication Award last year, which is the reason why he's here. This is where we find our excellence. He has received an Innovation and Best ECR presentation at Griffith Hack at the 10th International Nano Medicine Conference, and is also a guest lecturer for one of the Nano 2002 courses. So thank you very much, Nick. We look forward to your talk.
Thanks, Alessandro. That was actually a very nice introduction, and I actually seem somewhat professional, which is fantastic to be able to see it at such an early stage. So I'm just going to share my screen now. Hopefully that comes up for everybody. And as Alessandro has highlighted, there's two kind of main areas we're going to be kind of talking about today. First is an opportunity to meet me and ask questions of the work that we're particularly doing. And I also want to focus on the research that we are working on. So we look at nanotechnology for hepatic drug delivery, and we particularly focus on metabolic and age-associated conditions. So as an overview of what I hope you'll be able to get out of this session from a scientific standpoint is the broad benefits that nanotechnology can have for drug delivery. So this is going to be a mix between physics, chemistry, biology, clinical medicine potentially as well, and also its translation and how you kind of look at regulation for it. So what I hope to be able to show you is that the nanotechnology can have pharmacokinetic and biodistribution effects. It allows us to have a precision medicine that we're generating, how nanotechnology contains cellular uptake pathways, and also how a drug or what you're delivering can be better utilized by the cell or within an organ. The effectiveness of a drug or its pharmacodynamics can be greatly changed with nanotechnology, and I will demonstrate this with a few particular drug types. And the other thing we need to always address with nanotechnology is that toxicity component to it and how we want to evaluate that and the different approaches that can be done. So what I'm going to try and do is show you what we have done collectively over the last few years. And it's a broad ranging of techniques that are both nano and pharmaceutical related. And do feel free to ask questions as we go along. The other thing I do want to focus particularly with this talk is why we are looking at the liver. The liver is a remarkable organ. It's the center for metabolism in the body. It's key for most diabetic therapies and also key for how we do aging, particularly when we want to use drug treatments to target the aging process. And aging is one of the key risk factors for most disorders of which we get as we get older. So there's always a higher prevalence of us developing a disease as we age, and why aging is such a critical area to target. So a bit of an overview. That's me in the photo. We actually make some chocolate in the lab, and I can discuss that a little bit more later on. So I'm a lecturer, and usually people ask me straight out, well, what do you actually lecture on? So I usually say nanobiotechnology. I am a broad mix of different areas and expertise. So I'm based out of the Concord Clinical School, Concord Hospital at an institute called ANZAC. And the ANZAC focuses on aging research, so that's where we also come in. But then we've also branched into different areas. So we started looking at pharmaceuticals and nanotechnology, and that's kind of what's brought me into Sydney Nano. So I'm part of the Sydney Nano Health Initiative, particularly the Nano Pharma cluster that I chair with Pega, and that brings together how you pharmaceutically evaluate nanomaterials and how you can use nanotechnology for clinical translation. So feel free to ask questions about that as well at the end. The other thing I should highlight is I'm part of the early mid-career researchers subcommittee for the Faculty of Medicine and Health. So what we try and do with that committee is identify areas where we can try and improve early to mid-career translation, as well as support people during their early stage careers. So particularly on grants, we've got aspects we want to start looking at for supporting and also funding different types of early stage researchers. So as a tiny last bit of background just on my journey to where I've got to today. I did start off with a Bachelor of Science, a lot of different ologies. Pathology was my honours, physiology and immunology were my majors. I then changed to medicine and neuroscience for my PhD, and then following my PhD, I then moved on to pharmacology and aging type of research. And then this extended when we got an NHMRC grant for Victoria Cogger, that allowed us to investigate nanotechnology and the pharmacology of nanotechnology to try and target aging. And then we've gone from aging and then we've moved on to diabetes. So I'm going to try and go through a lot of that journey today and show you how we've come to this kind of stage. As a bit of a background, since I'm going to be jumping between physics, chemistry, and biology, where are nanomaterials used and how do you utilize them best within a biological setting? So nanoparticles are between the range of 1 to 100 nanometers. And my personal favorite nanoparticle is quantum dots for a lot of different properties, but mostly for their size, their fluorescence, and the fact that they can be used within a biological setting with relatively low toxicity. They can be water soluble and monodispersed, as we've shown here. So these are silver sulfide quantum dots that are about 5 nanometers in size. And you can control their sizing and also what they can be coated with. It allows them to be functionalized, and then we can use them for different roles and different pathways. The other key thing about quantum dots is they're very readily scavenged, particularly in the liver, and this aids their excretion. So it's something that can be given within an animal model, it'll be rapidly cleared, but you can deliver different materials to the liver. And the liver loves nanoparticles. The liver is the main problem for most nanotechnologists, and I'm sure everyone has been through this before. But since we target the liver, we love the fact that the liver loves nanoparticles because it's such a brilliant area to focus on. And lastly, quantum dots have broad-ranging activities, so they can be used for imaging, but also at the same time for transportation of materials and also for therapeutics. So I want to start off with a 24-hour life cycle of what happens if you've got quantum dots within an in vitro animal model. What particularly was known prior to when we published this work is if you give quantum dots, they'll be rapidly cleared within 24 hours in the feces. What we highlighted particularly within this paper is at the early stages of when you give them via oral administration, they don't just stay within the small intestine. They're transported from the small intestine into towards the liver, but they don't have systemic circulation, so they don't float around in blood. They're recognized by the liver, they're transported there with very high quantities. So this is over 60% of the amount of quantum dots we gave will pass through the liver. And that's a brilliant opportunity right there. And then they are slowly cleared over a 24-hour period. They didn't induce any toxicity with a single size aspect. And when you start looking at larger nanoparticles, so particles that are over 100 nanometers, they don't have this particular ability to go into the liver. We also did some histology to look whether or not we've actually got some retention, and we did go into more toxicity a little bit later on. So first attempt at playing videos. So this is the small intestine. So this is the area where the material that's in the small intestine, and these are blood vessels that will try and absorb different materials as they pass through. You can see our quantum dots are in green, small, so hopefully you can see as they slowly start moving, and there's continual green that does start perpetrating throughout the blood vessel in different streams. So this is actually quite rapid. So this is 15 minutes after we've given the materials to mice. So we've given them the gavage and then isolated out a part of their small bowel, and we're looking at that under a live imaging microscope.
Sorry, just as a first interruption, Nick, can you just clarify what is it we're supposed to be looking at? It's a little bit pixelated from where I'm sitting.
It's a little bit pixelated. If you do actually go to the publications, a lot more clear images.
Is it this green blob there that we're looking at?
So it's much more smaller particles. So they're probably these are 5 nanometer quantum dots. So really it is a pixel size, particularly this microscope. What we also have here is the blood vessels that are leading away from the small intestine. So again, it's green fluorescence with the quantum dots. And this is 15 minutes after we've been given the quantum dots.
Nick, it might be helpful to know what the length scales are.
Probably would have been a great idea, and they've disappeared from my figures. So this, the breadth of that's probably about 1 micrometer across for this blood vessel. So really you won't be able to clearly see quantum dots that are this size with this kind of microscopy. So what you'll be seeing is a green haze within this blood vessel, and that's our quantum dots. So it's just to show that we do have an imaging capacity to be able to try and see where these particles might be, but we don't actually have the resolution down to single particle level for these particular size quantum dots. The way you can actually do that is with transmission electron microscopy. So what we've done is we've given quantum dots and then looked within the liver 2 hours later. And what you can see within the blood vessels of the liver, particularly within the luminal space, is quantum dots that are bound to protein, which is usually referred to as the protein corona, and they have interactions with the cell membrane of the blood vessels that line the liver. So we've blown this up here in this section here. One of the key things I do want to highlight is within the lumen, so within the blood, these quantum dots cluster together within a protein corona. However, within these cell types, they are monodispersed, so they're not localized vesicles. What the liver endothelial cells, or the LSECs, do is these are the scavengers. So these will be the cells that are responsible for clearing nanoparticles from the body. They take them up, they recognize that they need to be transported to the bile, and then they're cleared rapidly from the liver. What we can also do is examine this within an in vitro setting. So what I've done is I've got quantum dots. I've then aggregated them together with BSA, and we can see as they interact with the cell membrane of this cell, is they will bind to the surface and then bits will be stripped off and it will be taken up into the cell. You can see down here the much more larger group has been interacted. So this allows us to look at the dynamics about how material is being taken up into cells, but allows us to look at what the in vitro aspect of this is. So we're seeing how these cells can be taken up into different cell types. And then we can look at different pathways that can be blocked. So we can pharmacologically block a particular uptake pathway and then see if that's responsible for why that particular particle is being taken up. The other key thing I want to highlight within an in vivo model is with nanoparticles, particularly quantum dots, there's a very high prevalence to be able to target the liver. But what this has been identified previously is a very mixed cell type that will take up nanoparticles in the liver. So it'll be a mix between immune cells, hepatocytes, and the liver endothelium that will take up nanoparticles, particularly quantum dots. And if you want, what is possible, if you functionally change the surface with a polymer that would be preferentially taken up by specific cells, you can change the dynamics of this uptake. So we can target the liver endothelium with 70% uptake and drastically reduce the immune uptake. We can target hepatocytes up to 80% and reduce the immune uptake. So you've got the ability to tune which cell type you can actually target. So now I want to touch on why we should want to target and look at the liver. The liver is an amazing organ. It is particularly looked at for anti-aging therapeutics that act on liver metabolism pathways. And liver dysfunction, as we both get old and from our diet, can greatly lead to metabolic disease, poor drug clearance, and impaired liver function. And this is most critical because we've got a prevalence to develop diabetes and metabolic syndrome. And diabetes and metabolic syndrome have the 5th highest mortality rate within the developed world. And they're the leading risk factors for cardiovascular disease, which has the highest mortality rate. So the fact that nanoparticles can target the liver with such high affinity is a brilliant clinical challenge that is currently being unmet. Cancer is probably the greatest area of nanoparticle focus, but it is the 4th highest mortality rate, whilst we have the potential to treat the highest in cardiovascular disease. Hence why the liver is so critically important. The other thing I want to highlight with this area is that different cell types within the liver have different functions and responsibilities. And as we even get older, they each have different problems that occur that can be addressed specifically with precision medicine by targeting these individual cell types. And this can be really important to develop better therapeutics. And it's why nanotechnology can be at the forefront for clinical translation. So I'm going to actually start working into some actual demonstrations of what you can do from a pharmaceutical point of view for our technology. So I'm going to give you 2 examples, and they're both medications for diabetes, metformin and insulin. As a broad biological introduction, they are materials that can regulate blood sugar, and we intend to utilize that aspect quite easily. So metformin can promote both glucose uptake and insulin sensitivity. And insulin, as everyone might be aware, is a very critical hormone that's required for treatment for all type 1 diabetics, and it's getting increasingly used by type 2 diabetics. So these are 2 medications that are within a very growing area of need that can be improved with the use of nanotechnology. And what we're looking at here is pharmacokinetic. So this is biodistribution, so where a nanoparticle will be. So what we have is radio-labeled metformin, and we're just looking at the location of it at different time points. So along here, and then relative to the amount that we've given them. So both metformin and our quantum dot version of metformin are orally bioavailable. But you can see here, there's a much faster uptake rate if you bind it to a quantum dot, shown by the one in red, compared to the one in black, which is metformin alone. And this is shown particularly in the small intestine, where it has a much slower uptake of absorbance, and you can increase this quite substantially with quantum dots. The difference between particularly at 2 hours can be up to 4-fold, just in terms of pharmacokinetics and biodistribution alone, to particularly target the liver. What I'm now going to look at is the pharmacodynamics. So this is the effect of the drug on a particular physiological action. So what I'm looking at is I'm giving metformin, which reduces blood sugar. You can see in a good dose response effect as it lowers blood glucose. You can see here with the dosage curve that we have here relative to its effect size. And then we've got our quantum dot version of metformin. And you can see it has, again, a dose response effect and a dose-specific effect size as well. The key difference between these 2 technologies is the dosages that we have to use. So we're using 25 times less drug in order to induce the same effect. And that's quite critical when we particularly start looking at the pharmacokinetic to pharmacodynamic effect size. So you can get a much more greater effect in a far faster time point when you start using nanoformulations of this particular type of medication. What I also want to highlight is we've changed how much of the drug you actually need to give, but does this actually change what the drug is actually there to be able to do? So this is the intracellular mechanism. So metformin will promote activation of phosphorylated AMPK, as well as STIRT1 and a nitric oxide specific molecule, pathway specific molecule, which is cyclic TMP. And you can see comparing black to the red, there's no difference between the drug alone versus the quantum dot formulation for the nitric oxide aspect in both the liver as a whole and within hepatocytes. But by changing how we're targeting this particular drug, we can get greater uptake in different cell types as well. So its effectiveness might be due to which cells it's being taken up into, but also how much of it is actually being delivered at that site. And you can also see when we've looked at other medications such as NMN, we also get similar types of effect sizes. So the last thing I do want to point out with a particular biologically focused cell type specific focus is we can change the cellular uptake pathways that are involved using nanoformulations. And this is probably one of the most key aspects of this publication we've just had. So metformin requires a very specific channel for its uptake, both from the small intestine, but also how it's utilized in the cells in the liver. And this is OCT1. And this particular channel can be blocked with protein pump inhibitors. So it's another medication that if patients that have type 2 diabetes are on, they have far less utilization of metformin because they can't take it up. It is also a genetic mutation that does affect people who also have type 2 diabetes. So they lose the effectiveness of the most common medication that's given for type 2 diabetes. And the key thing I want to highlight is when you use quantum dot formulations of it, not only do you get a difference in uptake rate from comparing metformin alone to the quantum dot formulation of it, is you're changing the uptake pathway. So you no longer need that OCT1 channel. So with our control group, with metformin, it's blocked by a protein pump inhibitor, osmopriol, but with the same inhibitor given to a quantum dot formulation in these cells, there's no effect. So you're changing it from needing an OCT1 pump to be able to pump it into the cell to then changing it to a catheter-mediated endocytosis, which is the most common form that cells can take up materials. So you're changing it from a very narrow specific area that the drug needs to work on to a much more broader way that it can get into the cell. But we've still seen that it's still utilized in the same way. So we're changing effectively the pharmacology of this drug with nanotechnology. And that's an amazing area that we can go into. So I do want to change gear from metformin and start talking about insulin. So insulin is a medication that is given via injection to all type 1 diabetics and increasingly to type 2 diabetics. It again regulates blood sugar. So it also has a effect on our blood glucose response curve in our oral glucose tolerance test. So we have an oral formulation with our quantum dot technology compared to the injectable formulation. So these have been given at the same time, so 15 minutes prior to the glucose test, and they both had the same effect on lowering blood glucose. Comparing an injectable treatment versus an oral treatment, we do need to use a higher dosage of the oral insulin, since we need more insulin to get in there, but it still can induce the same effect size on our glucose test, and this is the same area under the curve that occurs between these two treatments. The other key thing I want to highlight about using an oral formulation for insulin is that we are targeting the liver, and the liver is the single precision site that we're going for. Insulin acts on multiple areas. It acts in the liver, it acts in the muscle, and it acts in the fat. The key thing by using precision targeting to the liver is those other two sites are still able to perform their everyday function and regulate blood glucose. So what we see with our oral formulation is there's a reduced effect. So it has a maximal effect to be able to reduce blood sugar to similar to what a healthy dose of insulin can induce. However, if you increase the dosage of insulin that you're giving via an injection, you get a much more exacerbated effect. And this is quite dangerous because this is called hypoglycemia. It can occur in 15 to 20% of all injections, and in 1 to 2% of cases, it can require hospitalization. So hypoglycemia is a very large challenge, particularly for injectable insulins, and how you can address that both from the injectable side and the oral formulation side is a key challenge going forward for the area. And that's what our technology can potentially offer. So not only can it be an oral formulation that acts in 15 minutes with a reasonable amount of drug required, but it also has an added safety component of not inducing hypoglycemia in the models we've looked at so far. I do just want to touch on A lot of that data I just presented was from non-diabetic animals. So this is a diabetic animal model that requires insulin every day. What we've done is we're doing an insulin tolerance test. So this is either an injection or a gavage of our insulin formulations, and then we're just seeing the effect on their blood glucose. So they're both given at the same time frame, and within 15 minutes of the injectable insulin, you can get a reduction to a healthy level of blood sugar in these particular animals, and this effect can be generated with our oral insulin formulation. Again, there are dosage differences in the amount that we need to use. However, it's demonstrated it can induce the same effect. And this can occur in both early stage diabetes in these animals, moderate and also late stages. So it's the same effect that's occurring with insulin. And that goes again to what I've mentioned previously about it being utilized in the right way. It's being delivered, but also needs to be activating the same effect size. And the last aspect, and also most critically about this particular type of technology when you start want to be using nanoparticles, is its safety and toxicity. So what we've done here is we've looked at giving our silver sulfide quantum dots, the most toxic component of these medications, for 100 days with an everyday treatment. So our quantum dots are in their water and they continue to drink that for 100 days. So the dosage that I'm describing in this figure here is 10,000 times higher than that's being used for the oral insulin technology. So it's quite a high dosage. And what we observed was there was no change in body weight, no change in AST or LT, which is two key liver enzymes that highlight damage to both the endothelium and also hepatocytes. There was no indication of immune cell activation of interleukin-6 or interferon gamma. There was no indication of immune cell recruitment, which is with MINS2, which is very similar to interleukin-8. Sorry if it is just a whole bunch of different markers, but each one does have specific areas that are important for what they can kind of change and induce. What we did also look at is, since we're delivering all this material straight to the liver, we wanted to look at we were inducing any histological changes within the liver and also see if we're inducing any fibrosis. And there was no change between the fibrotic scoring that we saw in the liver and no change in their histology. We also did look at the kidneys, because this is another key area to look at for nanoparticles, particularly for their delivery. We saw no changes in their glomerular filtration, not glomerular filtration, their glomerular membrane, which is around here, which can be highlighted and gets much more larger and red when their filtration is impaired, and no change in their creatinine level, which is another indicator of kidney damage, particularly by nanomaterials. We also looked at TNF alpha as well and found no activation of that. So it's just to bring this all back to what we were discussing initially for this. I wanted to highlight that the benefits of nanotechnology, particularly for drug delivery, are to improve the pharmacodynamics and biodistribution. And although I focus particularly on the liver, you can use nanotechnology to target other areas of the body as well, with different degrees of affinity and precision. So nanotechnology has that ability to make sure you're delivering the drug to the right area, but in doing so, you also get the added benefit of this reduced side effects because it's not being delivered at other sites. So the main problem with side effects that we do see is when there's an effect that we don't want to have, but that drug still induces that effect, but in another area of the body. If we can target it to one particular area, we can reduce the incidence of that. And that's a fantastic area and particularly a very high clinical value going forward. What we've also shown is the cellular uptake pathways are tunable. You can choose which cellular endocytosis pathways are being taken up simply by functionalizing the materials. But what you also want to make sure that the drug is being utilized in the right way. You haven't changed how it's interacting. You haven't created a new drug. That's the other key challenge you want to be able to address. What you can also use nanotechnology for is greater pharmacodynamic effect, as well as the time frame of action for that activity. And you also want to ensure that you've got a non-toxic platform and utilization. The other thing I wanted to highlight from this talk was why we should focus on the liver. The liver is the metabolic hub of the body for maintaining homeostasis, and it is essential for maintaining that as we both get older and it's particularly under threat when we develop diabetic conditions. So all diabetic medications act on the liver, and greater effect on the liver can induce, in some cases such as insulin, reduced risk of side effects such as hyperglycemia. But you can also attach different types of medications with this platform I've described. I've only given examples of from metformin and insulin. However, there's a variety of different molecules you can have, and a lot of different drugs can be packaged within the same transporters at the same time to induce different effects. And you can also target that to different areas and different cells. So I do hope I haven't gone over time too much and everyone's still paying attention. But just some last minute thank yous, particularly for presented a lot of work, and a lot of far better people than me contributed to it. Particular thanks to Victoria and David, as well as Glenn at the ANZAC. We've also thanks to Wojciech and Christina from pharmacy at UCID. Sophie is now gone on to bigger and better things at NIH, as well as Jared, who's been hanging out with us and looking at it from a business specific area. So you can obviously appreciate that some areas of this technology can be translated clinically, and we're trying to get as much help on that as possible. Also want to thank Sydney Nano and the Sydney Nano Health Network and all of our co-chairs, and Ben as well, for their continual support and allowing us to better understand a multidisciplinary team and approach to better utilize this technology. And that's actually been really helpful. I do also want to acknowledge Zdenka as well, and Peter from Tromsø, and previous students in the lab have done a lot of the work, as well as funding from Australasian Diabetes Society, Haemophilia Foundation, Sydney Park, Spark, Drug Discovery Initiative, Faculty of Medicine Health, and the NHMRC. And I'm more than happy to answer any and all questions that you have. And thank you very much.